Water content or moisture content is the quantity of water content in a material such as soil. For many soil, the water content may be an extremely important index used for establish the relationship between the way a soil behaves and its properties. The objective of water content determination is to determine the water content of soil. For the moisture content experiment, we need soil sample, balance, moisture can and its lead. Firstly, we must make sure that moisture can and its lead is clean, so we wipe it with tissue. And we take the mass of moisture can and its lead. Next, we use around 10 to 20 gram soil sample and we take the mass of moisture can, leads and soil sample. After that, we must dry it under the sun. After we dry the soil sample, we take the data again. So, the difference between the mass of soil sample, moisture can and its leads after and before the drying process is the mass of water within the soil sample. So, to calculate the water content within the soil sample, the mass of water divided by the mass of solid and multiply with 100%. So, the result we get that the soil sample has 67.79% of water content. Even so, the data gain may be affected by the environment temperature. As precaution, the laboratory shall be conducted in the room temperature. Particle density is the weight of each soil particle per unit volume. Meanwhile, a ratio of the weight of a certain value of soil solid to the weight of an equivalent volume of distilled water at a constant temperature is called as specific gravity of soil. Specific gravity has range value for coarse soil, medium soil, and fine soil. The objective of particle density test is to determine the particle density and specific gravity of soil. There are three different tests for the particle density measurement. The first one is the density bottle method for a fine-grained soil. Next, gas jar method for soil that contain gravel-sized particle. And the last one is picometer method for medium and coarse-grained soil. For the particle density test, we need soil sample, density bottle and stopper, density water, vacuum, water bag, and balance. Firstly, make sure that the density bottle and stopper is clean. And we take the mass of both of them. We use around 10 to 20 gram soil sample and put it inside the density bottle. And we take the mass of soil sample, density bottle and stopper. After that, we use 10 ml distilled water and put it inside the density bottle. We shake and leave it around 2 to 10 hours. Then, we pour the distilled water until the half of the distilled bottle and put it inside the vacuum to remove the effect bubble air. Then, we pour until the distilled water is full with distilled water and put the stopper on. Then, put it inside the water bag for 1 hour. After that, we take out the distilled water from the water bag and dry it. Pour the water until the distilled bottle is full. Put the stopper on and take the mass. We empty and clean the distilled bottle and stopper. Fill it with distilled water until the bottle is full. Put the stopper on and take the mass. Again, we empty and clean the distilled bottle and repeat the same method for the sample number 2 and number 3. We put the data obtained from the experiment into the specific gravity formula, and we get that the soil sample has 2.20 specific gravity, which means that the soil sample is organic soil. Even so, there might be some error due to the remaining soil or water was still left inside the density bottle even after it was empty and used again. So, the precaution that can be done is to make sure to clean and dry the density bottle inside and outside properly before use again. Soil classification is a scheme for separating soil into broad groups, with each of them having potentially similar characteristics and behaviors. Soil can be classified into two major groups, which are coarse soil and fine soil. The objective of this laboratory is to determine the percentage of different grain size contained within the soil by using the sieve analysis technique. Involve several layers of sieve with different grade of sieve opening size. For the soil classification test, we use sieve analysis that need dry soil sample, sieve, and balance. Firstly, we use around 1 to 2 kg of dry soil sample and we take the mass of it. We also need to clean and take the mass of each of the sieve. Next, we pour the soil sample into the sieve that has been arranged according to the size of sieve opening. The largest one on top of the smallest one. And we put the lid on. We need to shake the soil around 15 minutes. After that, we take the mass of each of the sieve, including the butter pan, which contain of retaining soil. Next, we clean the sieve and do for stage number 2 and number 3. After that, we record the data and do some analysis by plotting the data on the graph. Finally, we can classify that the soil is well-graded very sandy gravel, referring to general identification and description of soil. Even so, there might be some error on the data since the cleanliness of the sieve has not been checked properly before the experiment started. Moreover, the value of the mass may be incorrectly transferred into the table. So, the precaution that can be taken is to make sure to clean the sieve from any other soil particle by using a suitable brush and always keep them in dry condition. In addition, check again the data obtained after transfer into the table and do the calculation carefully. Dry density of soil is defined as the ratio of mass of solid to the total value of the soil. It is a common measure of the amount of the compaction achieved during the construction. The objective of the laboratory is to determine dry density of in-situ soil from the soil that was excavated from the wet of sand by using sand replacement method. For the field density test, we will use sand replacement method that need sand, a set of porous cylinder, and balance. Firstly, we need to find the density of the calibration cylinder by using its mass and volume. Search for the suitable spot and excavate the soil using a trowel to a required depth. Place the sand porous cylinder centrally over the excavated hole. We can calculate the bound density of the in-situ soil and the water content of the soil removed from the hole by using the data obtained from the experiment. Finally, by using these two data, we can calculate the dry density of in-situ soil, which is 1.465 mg per meter cube. Even so, there might be some error during calibration process and the excavated hole was too small. As a precaution, make sure to use the right method of using porous cylinder. In addition, dig the hole into the soil deep enough and collect all the excavated soil to smooth the analysis process. Permeability is defined as the amount of water passed through the capacity of soil. Meanwhile, the coefficient of permeability is the flow of the velocity produced by the hydraulic gradient of unity. The objective for the following head test is to obtain the coefficient of permeability of fine grained soil. For the permeability test, we will use the following head test technique: soil sample, following head apparatus, water, cutter, marks, and tamping wood. Firstly, we will need to cut the upper part of the bottle to easy the process of put the soil sample inside it, and make hole below the bottle to make the water flow. After that, we put the soil sample into the bottle around the half of it. After that, we make 4 water level above the soil compaction. We put water until the bottle is full and reach the highest mark inside the bottle. We leave it for a while so that the soil sample can be fully saturated. Again, we add the water until it reaches the highest mark. By this time, we observe the time taken for the water to flow from H1 to H2, H2 to H3, H3 to H4. The same method was repeated for the test number 2, 3, 4, 5, 6 and 7. By using the data that have been recorded in the table, we may determine the coefficient of permeability of the soil sample. Even so, some correction need to be done on the result due to the effect of the temperature. So, the final result of coefficient of permeability of the soil sample that we get is 1.471 times 10 power of -6 meter per second. Even so, there might be some error during observing the movement of the water since the water flow quite fast. The precaution that can be taken is by repeating the test until they consistently produce the same result.